Okay, we're back for another fun-filled physiology, anatomy and physiology PowerPoint video. And hopefully what happens, you've already looked at the video before this, which is involving cardiac anatomy. That's going to help you a little bit here because there are some parts in the heart that you're going to have to understand where they are that will help you understand this. So if you haven't looked at that, please look at that first before we get to this portion where we talk a little about the physiology and how the heart actually generates a beat. And that's going to be sort of important down the road. So let's just talk about that in this particular discussion. First of all, when we look at the heart, okay, we know that there's the right atrium, left right ventricle, and all these other things like that. But within the muscle of the heart, there are basically two types of cells. One cells are called conducting cells, and we're going to be talking a lot in this PowerPoint video in regards to these conducting cells, how they work, where they go, what's the route, what's the pathway that creates a heartbeat. There are also the contractile cells, and the contractile cells we've talked about in the past in multiple areas in regards to cardiac muscle. Cardiac muscle is basically are going to be the cells that are going to cause the contraction. They're going to be the influence of the conducting cells, and we know they're very similar to skeletal muscle because they're striated, but one of the differences is they have multiple branches. So therefore what happens is that cardiac muscle can actually interact with a number of different cardiac muscle cells, which actually helps to propagate the electrical current and the contraction throughout the heart. Okay, let's just talk about what really happens in the heart. The heart's really sort of an interesting thing. Does the brain help to control the heart? Yes, it does, and we'll talk about that at the very end because, to be truthful with you, that's not where it all starts. Okay, the heart, what it does, it has its own generator. It's able to produce its own heartbeat without the influence of the brain or anything like that. And what happens, there's a place in the right atrium which is called the sinoatrial node, sometimes called the sinus node, or sometimes called the SA node. And this is an area, because of some special cells that are there, it actually creates what's called the action potential. And we've talked about the action potential in other situations, particularly when we did the muscle type of PowerPoint videos before. So this action potential is produced inside this this node of special cells in the right atrium. It's always in the right atrium. Okay, this is called the sinus node or sinoatrial node. And the thing about these cells that make them special is, remember, to start an action potential, we had to have some type of a stimulus that would allow those ion pores to open to allow sodium to come into the cell that changes that electrical potential on both sides of the cell membrane. Okay, what happens is these special cells in the sinoatrial node are particularly leaky. In other words, they let that sodium in a lot easier. In other words, there doesn't have to be some type of an outside stimulus. They're normally naturally leaky, which allows, just on its own, without any other stimulus, the sodium to rush inside, which then starts the beginning of this action potential that will be propagated along all the conducting cells, as well as being transmitted to the contractile cells. The other thing that's important to recognize, and we'll be talking a little bit more about in a few seconds, is the fact that once this SA node, sinoatrial node, starts with this stimulus and starts with this action potential there, what we find is that there's a regular pattern through which that electrical current is going to transmit itself through the heart, a very specific way. Okay, because what happens is the upper chambers, the atria, have to fill with blood as the ventricles are emptying. Then what happens is when the atria are filled, then what happens is the pressure in the ventricles goes down because there's nothing else, or there's less in there. So the atria now will contract and forces the blood from the atria into the ventricles. So now the ventricles fill, okay? And then what happens is then the ventricles are contracting to push things up while the atria is filling. So basically we have atrial filling or ventricular contraction, and atrial filling. So it's back and forth. So we have the atria contract and the ventricle, atria contract and the ventricle, atria contract and the ventricle. We can't have them both contract at the same time. Other than that, we have there's no pressure gradient for the blood to go. As well, you have the atrias working against the ventricles. In other words, the ventricle has to be, it's not always empty. There's always some blood. Probably about 40 to 50% of the blood still remains in the ventricle at the end of a contraction, but there should be some blood in the ventricle as a primer, but the atria has to have some place to put the blood, and so therefore there's a capacity that's still remaining in the ventricle, so when the atria contracts, the blood could go into the ventricle. Okay, so what happens is heartbeat, by this electrical current that starts at the SA node, has to go in a very specific way throughout the entire heart to make it a synchronous contraction. So the atria contract, then the ventricles, atria contract, then the ventricles, atria contract, then the ventricles, because the contraction forces blood out. The relaxation of the opposite part allows that chamber to fill, okay? And this electrical current that we see that's able to be transmitted throughout the heart is something that we could monitor by an electrocardiogram. When they put the electrodes on the surface of the body, we're able to detect that electrical current as it comes closer to the surface, or as it's transmitted to the surface of the body, and we're able to determine that. And by looking at it with an electrocardiogram from multiple different viewpoints, it could tell us a lot about the heart, okay? We won't get into electrocardiogram. I may make one a video just looking at electrocardiogram and how that comes to play, which would be extra, but because I think it's really interesting, it's a lot of fun to look at, but we won't be talking about that a whole lot right now. So this is what we're talking on the top, the little diagram on the top. What happens is we have the electrical stimulation starts at the AV node, and then it's going to travel through the rest of the heart to something called the across the atria, so the atria contract, then and also to the AV node, which holds that current up, then down through the ventricles, through the central wall of the ventricles, and then out through the outsides, which then allows the heart to contract from the lower portion to the bottom portion, and from bottom up. That way it forces blood up and out the way it should be, because the great vessels, as we talked about in anatomy, are on the base of the heart, or the top part of the heart. So I'll talk a little bit more about that process in a minute here, okay? So again, to review pretty much what we said with that cardiac physiology, the action potentials that we get in cardiac muscle are similar, but they're not exactly identical to that of skeletal muscle. And again, they are related to the same thing about that sodium rushing in and that change of polarity, because we know that in cells we generally have a more positive charge on the outside because of the presence, the higher abundance of sodium on the outside, and on the inside, the cell membrane, it's a little bit more electronegative, not because the potassium, which is in a higher concentration there, but because of large polar molecules and large negatively charged molecules like proteins and stuff that creates a net negative charge in the cytoplasm, or on the inside level of the membrane, okay? And so the idea, it all starts with, again, when that membrane becomes leaky enough, and in the SA node that's automatically leaky, to allow the sodium to rush in, which then starts, then eventually, we're going to have a situation where the potassium rushes out to try to meet things. We have calcium, which allows that contraction to occur, just very similar to what we had with skeletal muscle, okay? So the overall look at this, at the physiology of the heart, we know that, again, that the blood has to be driven from the atria of the ventricles, and the blood is driven from the ventricles out into the circulation, okay? And we'll talk a little bit more about that, which gets me down to the first definition that we have here, and that's a cardiac cycle. Cardiac cycle, whenever it's mentioned in physiology and whenever I mention it, means the beginning, the start of one heartbeat to the start of the next heartbeat, or the start of one a process where the atria contracts to the next one, okay? And we'll talk a little bit more about that as the time goes on. And this is what we see in what's called the action potential. Now we showed a very similar type of a situation with the action potential in skeletal muscle. And what we see, looking at our image up in here, okay, if I look here, this is right here what we call the threshold. This area right here is the threshold. And once I get leaky enough that for that to occur, we're going to have this upbeat, okay, and that's the area when that action potential is moving upward. That's the period of time when sodium is rushing inside the cell, okay. And then it takes a while for this, what's called, we'll talk about an absolute and a relative refractory period. During this period in here, okay, the heart can't be re-stimulated, and that's good. Simply because I mean, I can have really fast muscle contractions to make, but the heart can't do that. We don't want the heart to do that. Why? Because the heart has to relax enough to allow the chamber to fill. If there's not enough time for that chamber to fill, the heart doesn't have the blood to be able to pump out. We need to get the blood from one chamber to the other, and that's to allow, and the receiving chamber has to relax. This longer, what we call absolute refractory period, says the heart can't contract for a longer period in there, and that allows the heart to have a little chance to relax, to recover, and to allow that particular chamber to fill, okay? So if I'm looking, oops, let me get rid of that, get rid of all that, okay? And so this is just looking at an action potential and stuff like that that we see in a skeletal muscle. It's really short. Why? I could repeatedly fire it over and over again, as compared to a cardiac muscle where the action potential is spread out with this longer period, with that longer period in here, where what happens, it can't be really contracted. If I look in this yellow period right here, this is called the relative refractory period, relative refractory period, and basically that's a time when if I have a super high stimulus, the heart muscle may contract again, and sometimes that happens in abnormal heart rates. We'll talk about that a little bit later, okay? So what happens? Let's just talk a little bit more about how things happen. It all starts, like I mentioned, at the sinoatrial node, and that sinoatrial node, when we look at it, is that place right there that's circled. Okay, that's where the sinoatrial node is. It's sort of almost between where the superior vena cava and the inferior vena cava come together. That's where those magical cells are that are particularly leaky to let the sodium rush in without any other stimulus. Okay, another place that's really important to look at in the heart is what's called the atrioventricular node. Okay, and the atrioventricular node is this area right there. That's the atrioventricular node. And the atrioventricular node is sort of a gatekeeper. It's like the bouncer of the heart. What the atrioventricular node does, and we'll talk about it in a minute, it sort of holds that electrical current up just a little bit to allow the atria to contract before the ventricles. Okay, that'll be a little bit more explanatory. Okay, so we have the SA node and we have the AV node. What happens is these conducting cells, like I mentioned, are the ones that will conduct this electrical impulse in a direct pattern, in a direct route. Okay, so it connects the electrical signal between the two nodes, the SA node and the atrioventricular node. Also, we notice what happens, there are other pathways. There are a number of pathways in the atrium that allow that to be contracted, that allow that stimulus to be going from side to side, from the right atria to the left atria, and things like that. They're called intranodal pathways to the atrial muscles as well as to the AV node. And then the ventricle, we have a whole different set. So let me just show you what we're talking about in regards to that. Let's start with the SA node. Okay, we're up there. Those cells, like I mentioned, are leaky. So as a result, what happens on their own, without any stimulus from the outside, okay, at least normally, okay, what will happen is because they leak, that sodium will rush in, that causes the beginning of that action potential. When that action potential starts and we start to send that electrical current down, where does it go? What it does is it spreads all across the atria, from the right atria to the left atria, and it hits all the areas of the atria. Because I want to have the atria contract before the ventricles. Okay. At the same time, while it's contracting across the atria to cause the atria to become stimulated, it also sends another signal to the AV node, which I have pointed out here with the yellow. So I have that green arrow, which is an electrical signal that hits the AV node. Now that AV node says, wait a minute, hope it passports, I need to see your passport. It stops that electrical signal for just a little bit. Okay, that gives the atria enough time to contract. Okay, because I want to have the atria contract to fill the ventricles with blood before the ventricles contract. I don't want them contracting, like I said before, at the same time. At that point, once it's held up a certain amount of time at the AV node, it sends it down a bundle. And there's a little purple bundle that I drew in there, a little arrow. And that bundle is called the bundle of HIS, H-I-S, or atrioventricular bundle, AV bundle. And basically that's in the wall between the right ventricle and the left ventricle. That wall is called the interventricular septum. Septum is the wall between the ventricles. Okay, we have an interatrial septum between the atria as well, but it goes down the interventricular septum, and then what it does at a certain point, it spreads out. Part of it divides and goes into a branch that goes to the right ventricle, and part of it in this orange arrow goes to the left ventricle. So it divides, and those are called bundle branches. I have a right bundle branch and I have a left bundle branch. Nothing's happened in the ventricle so far. The ventricle hasn't contracted. This is just conducting the electrical current from that AV node through that bundle of HIS through the bundle branches. And it goes right down to the apex of the heart. And that apex of the heart we know is this area down here at that point. Okay, from that point, what happens is the electrical current takes a back turn. It goes backwards up from the apex towards the base, from the bottom to the top, and now as it goes up there are smaller fibers that come off these branches that actually are called Purkinje fibers, and these Purkinje fibers then stimulate all the contractile fibers in that area, which allows the ventricle to contract from the bottom upwards. And that's important because what happens is that I want to have that blood going from the ventricles out the aorta, out the pulmonary artery, because it's going, I want it to go upwards as compared to downwards. If the ventricle started to contract up in here as compared to contracting down here, I'd be pushing down this way. But when we have a situation where where, let me do it the wider line here. We have a situation where that electrical current comes down this way and this way. We contract from this point upwards, and it's actually forcing the blood upwards as well and out the great vessels, which is beautiful the way it's designed. It's a very nice way that that happens. Okay. Now, a couple things that I think are important. Oh, I misspelled heart there. What happens is the leakiness of the SA node says that this happens between about 80 to 100 times per minute. About 80 to 100 times per minute, it's leaky enough to let sodium come in to start this current going through the heart. Okay. If the SA node doesn't work and stops working, what happens is the AV node is a backup. You know, it's sort of like if I need it, I'll use it. And the AV node can have very similar cells that will do the same, but not as fast. And as a result, if the AV node, the atrioventricular node, controls the heart rate's only between 40 to 60, which might be enough to keep you alive, but you know, it's not good enough, okay, for a long run. So if the AV node is what runs it, because the SA node stops working, then the AV node, the heart rate's going to be less. If it turns out that the AV node itself is not working, then I have to rely upon Purkinje fibers, okay, and those Purkinje fibers basically are going to have a rate between 20 and 40, which is a rate that is really too low to do a whole lot. Sometimes if you look at a person whose heart's failing and is gone, what you'll see is every so often you'll see a little contraction here, you'll see a little contraction, they're called agonal, you'll see a little contraction there, little contraction there, and what that is basically the Purkinje fiber saying, you know, I got a little bit left, let me give it a try, but the SA node stops working. A lot of times what happens, you ever hear somebody they've had a pacemaker put in, and when someone has a pacemaker put in, it's usually because their SA node's not working very well, and what happens is they have a little box with a battery, and that battery will actually send a signal to the SA node to sort of push it to allow that sodium to come in, and that will help to control the heart rate. Okay, so basically what we find is the more damage to the heart, when I start to lose the SA node, my resting heart rate is going to go down. If my AV node is damaged and the SA node, I'm down to that Purkinje fiber level, which is between 20 and 40, which is far too little. Okay, so as I go to a lower site in the heart rate decreases in rate. Okay, now let's talk a little bit more about these individual nodes. Okay, again, that first node, that sinoatrial node or the sinus node, is on the posterior wall of the right atrium, near the superior vena cava entrance. The inferior vena cava comes up from the bottom, the superior vena cava comes from the top, and right by the superior vena cava is right where this SA node is. Again, it's known as the pacemaker. It's these pacemaker cells that are particularly leaky to sodium sit in that area of the SA node, and that's what in normal situations, probably all of you in the class, that's what actually runs your heart rate. Okay, it's the SA node that's doing this. Okay, it's the normal cardiac pacemaker in most people that have normal activity of their heart. Okay, and again, we talked before about how it connects to the AV node by these internodal pathways and the atrial walls and stuff like that. It takes about 50 milliseconds to go from the SA node to the AV node. I'm not going to actually have you remember that, but when we start to look at the overall time it takes for a heart to contract, we can actually divide that into, you know, the overall time, and we can actually see why the rate is what it is. So it takes about 50 seconds for that, or 50 milliseconds for that, but I don't think I'd remember that number. It's relatively rapid for that to occur. Okay, so this is step one. Step one, SA node activity begins where the SA node gets leaky, that sodium goes in, and as a result activation begins at that's time 0. Okay, and that takes us all the way up to where that stimulus spreads across the atria. Okay, so it's stimulating both atria, it's going through the wall of the atria and stimulating both walls of the atria. So now the atria will then start to contract. The elapsed time for this signal, this electrical signal, to go across the atria is about 50 milliseconds. Okay, about 50 milliseconds for it to occur. One thing I should mention is the wall between, let me write it in a different color and use a different way to do it. The area here, the wall between the atria and the ventricles, and the wall between the ventricles is thicker. It's almost like an insulating wall. And so as a result, the electrical activity that happens on one side of the heart is usually separated from the electrical activity that works on the other side of the heart. In fact, sometimes if we have a block in this area, which is called a bundle branch block, it's easy to see the effect on the electrocardiogram. Sometimes you'll see, let's say if they have what's called a right bundle branch block, you'll see the left ventricle contract on electrocardiogram, then you'll see the right ventricle contract. It almost looks like bunny ears. It'll look like that. You get this double peak where there should only be one peak. Okay. So that's jumping the gun a little bit. So now we know what happens is that stimulus has gone across the atria and electrical currents and spread across the atria. Okay, what happens is also part of that interatrial electrical flow allows that electrical current to get to the area of the AV node. Okay, and that atrioventricular node, AV node, sits sort of at the floor of the right atrium near the opening of the coronary sinus, which is the area where the veins that drain the heart muscle itself come into. It's right just above the level of the right ventricle. It's in the right atrium, the lower portion of the right atrium, just close to where it would start to go in through that wall to the right ventricle. Okay, again, that propagation rate is slowed at the AV node. It slows it up, it holds it up. It's the bouncer. You know, before they let more current in, they have to wait for a little bit. Fine, okay, you could go, hold it. You could go, hold it. You could go. And again, it takes about 100 milliseconds for the impulse to travel through the AV node and to get down to that AV bundle. I'll show you that in a second again, like we saw before. And again, the purpose of this AV node is to hold things up just long enough so the atria could contract while the ventricles are relaxed so the ventricles could fill. If the ventricles are contracting, I can't fill the ventricles. I'm forcing against a contracted ventricle and nothing works. So that's why we have that holding up at that AV node. That's a really important thing. Sometimes, if you ever heard of somebody with a heart block, okay, if you ever said, yeah, oh, grandma has a heart block, what happens is that heart blocks are usually a problem at that AV node where either it's held up too long or every other beat goes through or something that's wrong. It's usually a problem that occurs at the atrioventricular node when somebody has a heart block. Okay, and that's what we see with that. So let me get rid of these because I'm going to have you a little diagram. This little diagram shows right here, you know, as you can see, that now what happens is the atria is pretty much contracting, and we've gotten that signal down to the atrioventricular node. And what happens is the atrial contraction is going, okay? Atrial contraction is going, and everything's being held up, okay? So the atrial contraction is going, and everything's being held up at that AV node, so the ventricles can contract just a little bit later. Hope you get that point. It's about 150 milliseconds for that to occur. Okay. What happens is once the AV node says, okay, go on through, it lets the electrical signal on through the AV node, through that door, the AV node gets down into what's called the bundle of His. We talked about this before. It's called the bundle of His, or atrioventricular bundle. And basically, this is just a pathway, okay? And it goes in between. It goes through that interventricular septum, the wall between the right ventricle and the left ventricle, okay? So that's where it is, and it basically is where that electrical current that started in the atria is now going to be able to enter. Is now entering into the ventricle. Once it goes a little bit along that ventricular wall, it divides into a right bundle, like I mentioned, and a left bundle. The right bundle goes to the right ventricle, the left bundle goes to the left ventricle, okay? Because again, at that point, I have branches, and what happens on the right side doesn't really make that much of a difference on the left. What happens on the left side doesn't really make that much difference on the right, okay? So what will happen is the branches will then fan out, they go to the bottom of the heart, okay? I have a right bundle and a left bundle, they go to the bottom of the heart. Left bundle is going to go up the left side, right bundle is going to go up the right side. They're going to go to these things called Purkinje fibers that are coming out and branching out of these parts of the nerve, or the conducting cells that are up the wall of the ventricle, okay? So here's where we go. Here's what we see now in step 4, okay? In step 4 here, what we see in step 4 is we see now we have that current going down that atrioventricular bundle. Here would be the atrioventricular bundle. Here would be my right bundle branch. Here would be a left bundle branch. There's a little thing here called a moderator band, okay? And the moderator band is only in the right ventricle. I don't want to spend any time on it. In fact, I took it out of the PowerPoint presentation. But it's a shortcut across the right ventricle. And the reason why it's a shortcut across the right ventricle is if we remember back in our previous PowerPoint video on cardiac anatomy, if I looked at the muscle wall of the right ventricle as compared to the muscle wall of the left ventricle, the muscle wall of the right ventricle is thinner. It's not nearly as strong. So therefore, I want to give the right ventricle a little bit of advantage, okay, and let it contract just minutely, very small amount before the left ventricle, because once the left ventricle is saying, I'm the boss here, I'm going to take it all. So as a result, this moderator band, which is right here, allows a short circuit or a shortcut across the lower portion of the right ventricle, So that current could go up here, so it could start contracting a little bit before the left ventricle, and that's called the moderator bend. So when this actually happens down that bundle branch to get to the apex, and again we know that this down here at the bottom of the heart is called the apex. To get to the apex, it's about 175 elapsed time from the beginning of it to the end. So now we've gone about 175 milliseconds to go from when the SA node started to where it gets down to the apex of the heart. Once it gets down there, it goes to what we call the Purkinje fibers. I mentioned that as the conductile cells are coming up the side, they have these little branches that come out, and those little branches are called Purkinje fibers. Okay, and so what happens is the Purkinje fibers are stimulated by the bundle branches. That was a bad choice. Okay. The Purkinje fibers are stimulated by the bundle branch, okay? And therefore what happens is that as we go from the apex upwards, and these bundle branches become, the currents going up that, it actually stimulates the Purkinje fibers, okay? And this spreads throughout the ventricles, to the sides, to the front, to the back, everywhere else like that. And as a result, now we have a rapid conduction of that action potential along the ventricular muscle. And it actually covers all ventricular cells within about 75 milliseconds. About 75 milliseconds to go from the apex up to the sides. So as a result, now what's going to happen is I'm going to be able to contract. This is another thing. I want to have the atria contract before the ventricles. But here's another cool thing. By having nothing happen until we get that, until those bundle branches get down to the apex, and then the contraction starts when it starts to make that turn going back north by all these Purkinje fibers. That means that the apex will also contract a little bit before up in here. So we have the apex contracting a little bit before the part of the ventricle up here, which is good. Why? Because those great vessels are on the top of the heart at the base. So if I contract from the bottom up, what happens? I'm actually pushing the blood from the bottom up, which is the direction it has to go. Absolutely perfect. Okay, so that's what works out with that. That's why the heart does what it's supposed to do. Okay, and so if I take that 75 milliseconds and add it all, the whole time for this to happen, the whole elapsed time to get to here where I'm getting this contraction of the ventricle is about 225 milliseconds. 225 milliseconds. Okay, so that again, don't remember the time, but that's like just an idea about the time. Now a couple things, and then what happens is then we start that whole process again. After the heart contracts, after the atria contracts and the ventricle contracts, I need a time for that atria to relax, to fill. So there's always a little bit of a relaxation period after that contraction. And you got to remember that when I looked at that action potential of a cardiac muscle, we had that high uptake, and then we had that long area where we have what's called an absolute refractory period out here, where the muscle can't be contracted. During that period of time, it's relaxed, okay? And that allows that chamber to fill. It's a horrible relax, okay? But you know that means relax, okay? Now, a couple things I want to mention here that I think are important about this heart conduction. First of all, the heart can actually contract and conduct impulses to a maximal rate of about 230 beats per minute, which is a lot. It can't go much higher than that. I know with me, I have a defibrillator in my chest. I have a box, okay? And it's gone off 3 times when my heart went into this ventricular fibrillation. It's gone off 3 times, and the one time I got a strip, I should get that sometime and just print it out and show you. One time the strip was up to about the contractions were up to about 220 beats per minute, okay? And that's really getting almost maxing out. The heart's going so fast. The thing that's important, okay, and just think about this logically. Don't even think about it physiologically, but think about it logically. For something to fill, it needs time. So the faster the heart rate, it can't fill fast enough. The ventricle, to be able to pump out to the vessels, can't fill fast enough. There's just not enough time between contraction to contraction to contraction. Once I get up to 180 beats per minute, that what happens is there's not enough time to fill, and if the ventricles can't fill before they contract again, there's not much blood coming out. Makes sense. Once I get up really high, to 200s, basically, effectively, there's almost no circulation going at all. Why? Because I can't get the ventricles filled enough to be able to pump blood out. And I need that time, that relaxation time between contractions with that absolute refractory period, to be able to let the ventricle and as well as the atria to fill, so it could fill the next chamber. In other words, atria filling the ventricles, and the ventricles sending blood up out into the vessels, as we talked about in the cardiac anatomy. Okay, usually when somebody gets a rate that's over 230 beats per minute, somewhere in that range, it's most likely because some type of a damage to the conduction system. With me, when I was 37 or 36, okay, I had a myocardial infarction, which is a heart attack, and what happens is when people have a myocardial infarction, the part of the heart that gets infarcted, okay, which is basically damaged because the blood supply periodically goes down, that part of the muscle will die. That muscle is not going to come back. It never comes back. Once it's gone, it's gone. So what it does, it's replaced by scar. That wall there gets replaced by scar. Well, guess what? Electrical impulses can't go through scar. They have to go around. Okay? And in my situation, what was happening is the scar, which is actually quite large, the electrical impulse comes down and starts to go on a traffic circle around the scar, and it passes where it goes around, and it re-excites itself. It's like a dog chasing its tail, and as a result, that's what makes that heart rate sometimes go way up. Okay, there are other things that may do it as well, but we'll talk about that in a little bit later, about some other hormonal influences that'll make the heart rate go up. But again, if we usually have a rate that's over 200 and some beats per minute, it's usually because there's some type of a damage or defect to that conduction, that electrical conduction system in the heart. Okay, so that's what happens with that. The entire time, the entire process, therefore, when you looked at all those times, to go from the SA node, sinoatrial node, to the Purkinje fibers was about 225 milliseconds. Okay, and the idea is with that AV node there holding things up, it could have been shorter, but because that AV node holds things up and lengthens that time, it then allows the atria to contract before we have depolarization of the ventricle. Okay, depolarization means that the electrical current's gone and depolarized it so it could start to contract. Okay, and again, like I mentioned before, the Purkinje fibers radiate from the apex, the point of the heart, upwards, and that way the contraction of the apex before the base, so it pushes up the blood upward out through the pulmonary artery as well as out through the aorta. Okay, pulmonary artery from the right side of the heart, aorta from the left side of the heart, which is all very advantageous. Okay, and it all works out really well. Okay, step 5, again, the impulse to distribute our Purkinje fibers to the thing. So there's my 225 milliseconds, like we talked about. Okay, now again, the thing that's interesting about this, and we sort of touched on it before, is there's what's called an automaticity, okay, or autorhythmicity of the heart, because again, those specialized cells in the SA node, because they're made to be leaky, okay, they initiate and distribute the electrical impulses without any other influence. They can be influenced by other things such as the vagus nerve, which slows it down, or other sympathetic nerves which may speed it up and stuff like that. But what happens is these cells, because they are made special, and they're special cells in the sinoatrial node that are leaky, will allow this to start without any other thing automatically. If you remember, like I mentioned in the previous video, is, you know, Indiana Jones' Temple of Doom, that priest reaches in that guy's heart, chest, and pulls out his heart, and his heart keeps on pumping. If you take a heart out of something and put it on something, it will continue to beat for a while until it actually runs out of all those chemical mediators that allow that to occur. Okay, and where it runs out the energy, because what happens is, remember, if sodium rushes in and potassium rushes out, now I have potassium on the outside, sodium on the inside. Whoop, that's wrong. I have to get this potassium back in and sodium back out. It takes a pump to do that. What does that pump require? Energy. Eventually that energy is going to run out, and I can't reverse it, and therefore that contraction, that whole process will stop. Okay, a couple other things. One other thing I should mention is that the contraction that we see of the muscle is slightly behind the conduction, so the conduction goes through a little faster than the contraction. And the reason why this happens is, when we talked about skeletal muscle, we talked about how the electrical potential, the action potential, went along the sarcolemma and went down the end of the sarcomere, and it allowed calcium to be released in the sarcoplasm, which then bound to the troponin that allowed the tropomyosin to start to pull. Okay, well, guess what? Calcium is important in the heart too. So what happens is that contraction is a little bit, because if I could stimulate it, but it takes a while for that stimulation to go along the surface of that conducting cell to be able to, and the contractile cell, to be allowed to let the calcium rush in to cause interaction of myofilaments in the heart to be able to allow that contraction to occur. So there's a slight delay slight between the contraction and the conduction. So in other words, the contraction's a little bit behind that electrical current goes through, and that's because that calcium has to be released. That calcium has to do its magic in all the myofilaments in the heart muscle to allow that contraction to occur. And that's what happens with that. There's a little bit of a delay. Now let me just mention cardiac cycle, okay? Cardiac cycle is what happens is if I look at the heartbeat, I have a heartbeat followed by a brief rest period, and the chambers relax. In other words, we have atria, ventricle, and they both relax a little bit. Atria, ventricle, little relax. 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 Atria, ventricle, little relax, and stuff like that. And that relaxation period allows for the next heartbeat to go, and it sort of gets things back to normal. And so therefore, that gets me to what we call the cardiac cycle. The cardiac cycle, by definition, is the beginning of one heartbeat to the beginning of the next. Okay, we talked about that earlier. Now, two words that are essential. Two words that are essential. Next time you want to get a tattoo, this is what you need to have put in your tattoo. One is systole, and the other one's diastole. Systole means contraction. Okay, and most of the time in the heart, we talk about ventricular systole because that's the most important thing. And basically, systole is the contraction or the pumping of the ventricle. Okay, where the pumping chambers are forcing blood out. Diastole is the relaxation or relaxing period or filling time of the ventricles when the ventricles are filling. Okay, so when you hear the word systole and diastole, systole means the heart, the ventricles are pumping and pushing blood out. Diastole means the ventricles are relaxing and allowing the chambers to fill. When you take your blood pressure, you get like, let's say 120 over 80, yeah, but they say, even though it's not. When they say 120 over 80, 120 stands for what's called the systolic pressure. 80 stands for the diastolic pressure. Basically, that systolic pressure is sort of measuring the pressure, the pumping pressure of inside the artery as the ventricles are pushing the blood forward, and that's what that measures. So when you get that higher number, it's that pumping pressure as the ventricles are forcing blood out through the heart and into the vessels, and that pulses or that pressure is appreciated. The diastolic pressure is the lower pressure, and that means that there's always some pressure in the vessels. Why? Because even when the ventricles contract, they eject probably about 60%, maybe 70% of the blood in the chambers. There's always some blood in the ventricle. There always has to be. The pump always has to be primed, okay? So because there's always some fluid in the vessels, it says that that gives me a lower pressure, which is called my diastolic pressure, okay? So those are what those two pressures mean. Let's go here. So what happens is we also have to recognize that fluids run from move and flow and run from a higher pressure to lower pressure. Okay. Now, when the ventricles are relaxing, the pressure inside the ventricle is lower than the pressure in the atria. Why? Because the atria is full. The atria is full of blood, so therefore the pressure, because now that atria is expanded and full, is greater. So therefore what's going to happen is the valve between the atria and the ventricle, on the right side, that tricuspid valve, on the left side, that bicuspid or mitral valve, will open to allow the blood to go into the ventricles, because the pressure is going from a lower pressure to a higher pressure. Okay. Again, the pressures increase with systole and decrease with diastole, as we talked about. And as we talked about in the cardiac anatomy, is that those valves allow one-way flow only. So when the pressure in the atria is greater than the pressure in the ventricle, those valves will open. Now all of a sudden, and because the ventricle is relaxed, now that the ventricle opens, the blood now, the atria will contract, squeeze, and now the blood's going to go into the ventricles. Okay, now the atria is relaxed, so what will happen is the valve will close because of these. We talked about the papillary muscles and the chordate tendon will actually cause that valve to snap shut, whether it be the tricuspid or the bicuspid valve, and therefore when the ventricle contracts, it's going to go out the other valve into the aorta or the pulmonary artery, which now has less pressure than what's in the ventricle, because it's going from higher pressure in the ventricle now out through the aorta or out through the pulmonary artery. Okay, and so that's what happens. And again, the whole key to all this is what we talked about in the first 20-some minutes here, is the fact that that pacemaker in the conduction system of the heart makes this happen. In other words, it synchronized it to such a fine degree that allows my the blood that's coming out of my heart to come out in a very effective and efficient way. Okay, and again, the atria determines that normalcy by how the SA word works. If the SA node doesn't work, then I have a problem. Okay. at the beginning of the cycle, all chambers are relaxed and the ventricles are partly filled. Like I said, the ventricles always have a little bit of blood in them. They're always a little bit primed with blood, okay? They eject a certain amount of blood with each contraction, and they call that the ejection fraction. The ejection fraction. And the ejection fraction basically is the percentage of blood that actually will be pumped out at each systole, each ventricular systole. The amount of blood that's pumped out is called the ejection fraction, which is usually between about 50 to 60%, maybe even up to 70%. Now, blood's pushed into the ventricles, takes about 100 milliseconds. Atrial pressure is greater than ventricular pressure. Therefore, what happens is minimal backflow, and what happens in atrial systole, blood goes from the atria into the ventricles, just like I mentioned before. This is a review of what we said, okay? Blood follows the path of least resistance, higher pressure in the filled atria, lower pressure in the relaxed and less filled ventricle. So therefore, when the atria contracts, the blood's going to go through that tricuspid and bicuspid valve into the ventricle. And that's what we see with atrial systole. And that's what we see there. And you can read that over. Atrial systole begins. Atrial contraction forces a small amount of blood, a small amount of additional blood into the relaxed ventricles. Boom. Okay? That's what we have. Then we go to atrial diastole. Now the atria is going to be relaxing and filling at the time that ventricles are going through systole. Okay, and everybody knows systole and diastole. The atrial diastole continues until the next atrial systole, so it's filling. It'll gradually fill. Okay, for that time it's relaxed. Okay, and what happens is that atrial diastole begins about the same time as ventricular systole. Just when the ventricles are pumping, the atrials have finally said, okay, it's time to relax, and they start to fill. Okay, and that takes about 270 milliseconds. Okay, blood's forced from the ventricles in the pulmonary artery on the right side or the aorta on the left side from the ventricle as the atria is relaxed. So that's ventricular systole, which is what we think about the most. Okay, and now we're talking about right here. Atrial systole ends and atrial diastole begins at that point, and then finally get down there where ventricular systole begins, the first phase, which actually forces blood out through the area of the because the pressure now is increased with all the extra blood in the ventricles. The muscles contract because now the electrical current comes down, gets to the apex, and starts to go up and starts to push the blood from the apex upwards. And that's what we see in this image right here. Then we go into ventricular diastole. Once the ventricles have pumped out the blood that they're supposed to, then what happens is the ventricle has to relax. It has to fill. And that takes a while. It takes about 530 milliseconds. Okay? It takes a while for that ventricle to fill. And that's when both the atria and that ventricle are sort of in a period of relaxation together. Okay? So near the end of ventricular diastole, atrial systole starts, and then we start again. Okay? And we repeat this process over and over and over, normally between 60 and 100 times per minute, every minute of every hour of every day of every week of every month of every year. Okay, and that's what we have with that. And there we are, where we have ventricular diastole. It's filling at that point. We start to fill it up, and finally we get further diastole, where all the chambers are relaxed. Ventricles fill very passively. There's nothing that sucks the blood in. It's basically coming in simply because the pressure in the ventricle, when it's less full than the atria, will allow the blood to go from the atria into the ventricles. Okay. Again, we're talking about that cardiac cycle. Again, we talked about this before, is that if I have an increased heart rate, the faster the heart rate goes, the less time there is between systole. Okay, and therefore the ventricles don't have a long enough diastole to be able to fill. That decreases effective blood flow. Okay, probably has a greater effect on decreasing time in diastole because the chamber can't fill. If the chamber can't fill, there's nothing to pump. Okay, it fills a little bit, but doesn't pump enough, or doesn't fill enough to be able to pump out enough blood that's going to be conducive to a good, happy life. Okay, so if the heart rate's too fast, the ventricles don't have enough time to fill. That's the crux of it all. And the problem is because of the problem with filling the chambers. Okay, and what happens is, therefore, if I have a shorter period of time, the amount of blood that's able to be ejected at each systole of the ventricle becomes less. It goes down. Okay, so we have a lower amount of with each systole. Okay, now this brings me to another concept and something else that you need to know. You definitely need to know this word: cardiac output. Cardiac output is the amount of blood that the heart pumps by each ventricle per minute. Okay, it's anywhere between 5 to 6 liters of blood that's moved per minute. Okay. So the cardiac output is the amount of blood that's pumped by each ventricle per minute. Okay, And what happens is we determine this cardiac output by two things, by two factors, okay? If you think that the blood has to come out, what influences blood coming out of the ventricle? Number one, the rate. Let me do a different color. The rate. How fast the heart's going, okay? That's an important factor. Second thing is the amount of blood that's being pumped out. If I take the rate times the amount, that means my cardiac output, okay? And the way we get that is the heartbeats per minute times the stroke volume, which is the amount out, gets me my cardiac output, okay? So the cardiac output could be calculated by if I know how fast the heart rate is and I know how much blood the heart's pumping out each beat, I could determine the total amount of blood that's going out through the heart per minute. Makes sense, okay? It's logical. So if I take the heart rate and multiply that and consider the stroke volume, which again is let me get rid of all this so you can actually see. Let me write a couple other things in here. Got a lot of stuff to erase here. If I take the heart rate, okay, over here, if I take the heart rate and I multiply that by the stroke volume, which is the amount out per systole, that gets me how much is coming out per minute. So my heart rate is like, let's say it's, so if my heart rate was 60 beats per minute, I would multiply the 60 times how much is being ejected with each beat, and that gets me how much blood's being sent out of my heart per minute. So the cardiac output is in liters per minute, okay? Couple other terms that you might want to know, okay? And this also influences these cardiac dynamics we talked about, okay? First thing we have is what's called end diastolic volume or EDV, okay? The end diastolic volume, let's say I have 100 cc's of blood, just as a round number, 100 cc's of blood in the left ventricle, okay? And if I pump out, if my stroke volume is 60 cc's, at the end of systole, okay, what would that be? That's my end systolic volume. So at the end of systole, after I pump it out, if I have 100 cc's, which would be my end diastolic volume, okay, so 100 cc's, let's just take that as an example. I start out with 100 cc's. At the end of the time when the ventricle is totally filled, I have 100 cc's of blood in there. That's at the end of diastole. What's diastole? The relaxation or filling period. So if I have 100 cc's, okay, then what happens is at the end of systole, let's say I pump out 60, my end systolic volume is 40 cc's. Make sense? So 100 minus 60 is 40. So my end systolic volume is the amount of blood remaining in the ventricles at the end of ventricular systole. So again, if I start out at the end of ventricular diastole when the ventricle is filled with 100 cc's, and I eject, my stroke volume is 60, send out 60 cc's, that means at the end of systole, I still have 40 cc's left inside the ventricle, okay? This also could be made into a fraction, okay? If I take the 60 cc's and put that as a fraction over 100, this is right here is called my ejection fraction. So you might see EF, hard to see right there, EF. EF means ejection fraction. I'm ejecting 60% of my blood out per each contraction, okay? That's pretty, you think about it, it makes sense. If the heart has 100 cc's in it at the end of diastole when it's totally filled, and the heart pumps out 60 cc's, it means it's pumped out 60% of its volume. That 60% of its volume is called my ejection fraction, which means that I have 40 cc's still left inside the ventricle, and that's called my end diastolic volume, okay? Or end systolic volume. So again, if I look at some of this cardiac dynamics, stroke volume is the amount of blood that's pumped out of each ventricle with each beat. So stroke volume equals the end diastolic volume minus my end systolic volume, okay? So if I look at my end, let me get back here. If my end diastolic volume is, I'm just going in reverse. If my end diastolic volume is 100 cc's and my end systolic volume is 40 cc's, how much have I ejected out? I've ejected out 60 cc's, and 60 cc's is my stroke volume. Just we've done it in reverse. And again, if I look at that ejection fraction, that's the percentage. So if I take this percentage, I've ejected 60 cc's out, 60 cc's over 100, is 60%. Okay, so normally that ejection fraction is somewhere between 50 and 60% in normal people. I know with me, with my past myocardial infarction, my ejection fraction is about 30%. Okay, it's not really very good, but hey, you live with what you got, So that's a couple other things. And again, if I look at my end diastolic volume, 100 cc's, end systolic volume, 40 cc's, my stroke volume would be that 60 cc's. So I might have, even on the next exam, calculate the stroke volume for the following numbers, and you should be able to do that. And it's pretty simple. Again, if I know that I have 100 cc's in my ventricle at the end of diastole, it's totally full. And if I eject blood out, and at the end of systole, end of that ventricular contraction, I have 40 cc's left. That tells me I ejected out 60 cc's. If I take that 60 cc's and multiply it by heart rate, that gets me my cardiac output. One thing leads to the next. And if I want to determine an ejection fraction, if I think I've ejected out 60 cc's when I started out with 100, then I'm ejecting out 60% of the blood is being ejected from the heart with each heartbeat. So I'm able to calculate those things. Now, one last thing before I tie this one up at the end, there are a lot of factors that affect the heart rate, and I'm going to be actually putting together a very short video in the next day or so in regards to some of the other factors or some of the other things that happen in the heart that will affect. But what happens, you got to think about a couple things. Factors that affect the heart rate. First of all, again, let's go back and think about what we talked in the last section, the nervous system section, that what happens is that my medulla oblongata really does a lot of control of my heart rate. We have a lot of autonomic activity, and you should know what autonomic activity is. We have a lot of autonomic activity because of the medulla oblongata, okay? And it's firing. It could either be parasympathetic. What do you think parasympathetic activity does to the heart? It slows it down. Again, remember, parasympathetic activity has a tendency to slow things down, with the exception of the GI tract, where sympathetic activity has a tendency to speed things up. Sympathetic activity makes things go faster. Okay, so as a result, when you get scared or you get angry, as your heart rate starts to go up. Why? Because now the medulla oblongata says, "Hey, we need to push the envelope here a little bit. We have to go a little bit faster, a little bit harder. We need to." It's that fight or flight type thing where if you're getting scared, you want to run. And you run faster. Why? Because the medulla oblongata sends out sympathetic stimulus to the area of the heart that will increase the heart rate. What does the increased heart rate do? It circulates more blood through the rest of the body until it gets to a certain level, obviously. Circulates more blood, because the body is going to need more blood with more oxygen for more energy, because I'm going to need more energy to run away. Really simple. Also, what's going to happen is that because of that sympathetic activity, it may affect what's called the adrenal gland. The adrenal gland or suprarenal gland are two little glands, one on top of the right kidney, one on top of the left kidney. And one of the things that the adrenal gland, or two things that the adrenal gland make, are epinephrine or adrenaline and norepinephrine or noradrenaline. What these are, they're called sympathomimetic materials. Okay, they're actually hormones, sympathomimetics. What does it mean? Sympatho means speed things up. So these hormones then get released in the adrenal and makes things go faster. So we do get some hormonal activity from the adrenals with the epinephrine, adrenaline, or norepinephrine, noradrenaline that will occur, and they will affect the rate of the heart. Again, that parasympathetic, we talked about the vagus, how when the vagus gets stimulated, the heart rate goes down, starts to slow down. Okay. So what do we have that involves the heart rate? Again, autonomic innervation, largely from medulla oblongata, sympathetic and parasympathetic, as well as hormones. And some of those hormones largely come from the area of the adrenal, the epinephrine and norepinephrine. There are other ones that will make it go faster and stuff like that, but that's what we'll talk about right now. So the sympathetic and parasympathetic fibers are in what's called a cardiac plexus. Around the area of the heart, there are a bunch of nerves, and because we know autonomic nerves, we have all kinds. They're a mix, okay? Sympathetic, parasympathetic fibers, they're all there. And again, the most important parasympathetic fiber that we have going to the heart is that vagus nerve, which is primarily is basically parasympathetic. Okay, and then and again we know that that's in the medulla oblongata, like I just said a minute ago, and that's autonomic control. I don't really control that. In other words, if you get scared, your heart rate's going to go faster. You're not making the heart rate go faster because you want it to go faster, but it is because something happens, and that's the medulla oblongata says, hey, get moving, Buster. We got to get out of here. Okay, so those are inside the medulla oblongata, they have a cardio-acceleratory center. What do you think that does? Makes it go faster. So it's sympathetic, it increases the heart rate, and we have a cardio-inhibitory center, which is basically parasympathetic, which makes the heart rate go down. So it slows the heart rate down. Okay, and that's what we see, with some of these